And so we are isolating proteins. All right. Up to this point in time, we've used salting out, we've used dialysis, and we've used chromatography. Techniques, also centrifugation, techniques to isolate very large amounts of active protein so that we can study. What we're now going to do is use electrophoresis, and we use some electrophoresis in terms of isoelectric focusing to separate proteins, very small amounts, very fine resolution, so we can really get separate proteins in terms of very small differences. Now, isoelectric focusing is interesting. It's not used as often as this technique, the regular gel electrophoresis that's coupled to with the use of sodium dodecadyl sulfate, or SDS. What we're going to use is electricity to move things, right? But the electricity doesn't separate anything. It's the jello, or the gel that we go through, that separates. Okay, and so what we're going to do is we're going to polymerize a gel made up of acrylamide and bisacrylamide, and the combination of those two components, if I add the bisacrylamide in higher concentrations, what I'm going to get is a thicker gel that has smaller pores. If I use less, I get a bigger gel. And so I can have a lot of screening effect or a little bit of screening effect. And so when I polymerize them, it is called polyacrylamide gel electrophoresis, or PAGE. That's where that name comes from. Now, we used to actually allow our students to make polyacrylamide gels, but the problem is the acrylamide is a neurotoxin, and so you had to be careful with it. And so we decided, no, let's not do this with the undergrads. And so we buy the gels now. But if you're in grad school, the number of gels that you use, you will be using the polyacrylamide. You'll be making your own gels. The idea here is we're going to separate by size. And as we move the molecules through the gel, here's what happens. Small ones can get through faster than big ones. All right. We use chromatography, right? Separate by size. What came out the bottom? Which size came out first? The big ones came out first. Small ones got hung up in all of the little holes. The big ones came out first because they didn't have to actually enter the holes. They fell around the beads. Here, in acrylamide gel, I have to have every protein go through the screening effect. So the smaller ones can get through the screens faster than the big ones. The big ones get hung up. This is probably where most people get confused. We're separating by size in two different techniques, and they do by two different ways, methods. And so knowing which one moves faster in which process is important. All right. Now, when we do this, we not only move them, but they get trapped inside the Jello mix. And what we tend not to do is move the proteins all the way out. We're going to trap them in the matrix and then analyze them from there, either directly in the gel, or we'll transfer them and analyze them in a different way. Very high resolution. The amount of screening is controlled by the percentage of the cross-linker, which is the bisacrylamide. Now, here's the problem. Did you see that gel? I had all the proteins at the top and all the proteins at the bottom, right? How do I make all the proteins run in one direction? In isoelectric focusing, did they all run in one direction? No, because every protein had a different charge, right? And if it was positive, it went to the negative electrode. If it was negative, it went to the positive electrode. And as it reached a different pH, then the pKa changes and the protonate stops when it gets neutral, okay? Well, proteins have all these different charges. But I want to make all the proteins run in one direction. And I'm going to make them all run towards the positive electrode, okay? But not all the proteins are negative to run towards that positive electrode. So what I have to do is I have to mess up the protein. And what I do is I am going to denature the protein using the detergent called SDS, sodium dodecadyl sulfate. All right, what is denature? unfold it. I'm going to unfold it, and I'm going to use a particular detergent called sodium dodecadyl sulfate. Okay, what do you know about any of those words in sodium dodecadyl sulfate? Do you recognize any of those words? Sodium, got it. Do you know what dodecadyl is? Dodec means 12, and we're going to get there. And then what else? Sulfate. Okay. Do you know sulfate? Okay. Feel it. Feel sulfate in your soul, right? You could write that ion on the board, right? What charge does the sulfate have? -2. And if I asked you to draw the Lewis structure, could you do that? Showing the formal charge on each atom. I make my freshmen do it. You guys are past freshmen, right? Yeah. Okay. What's important? All right. The fact that it has the sulfate -2 charge is what's going to overwhelm the charge of the natural protein state. Now, the dodecadyl is important too. Long hydrophobic chain. When a protein folds, what goes on the inside? What amino acids are on the inside? Very hydrophobic. What is this part of the molecule right here? It's very what? This part right here. Very hydrophobic, right? So what's going to do is the dodecadyl sulfate, the actual hydrophobic chain, will match to the backbone, really, of the peptide. And it will bind in there, disrupt the internal hydrophobic, coat it, and then on the outside put a negative charge. And what happens, about every 2 amino acids will take up one of these SDS. And so if you have 100 amino acids, then you'll get 500 of these sulfates on there, and it will be negatively charged by a lot. If you had 1,000, then you'll have 500 of these on there. And so what you do is you make essentially the same mass to charge ratio, and it will be overwhelmingly negative. Even it overwhelms all the charges that are there on the original protein, and it makes them overwhelmingly negative. Now, it's not absolute, but it's enough. Okay? Now, if every protein had the same charge to mass ratio, if you put those proteins in a solution that had electrodes, and it just has the buffer, no screening, all of the proteins would move through the buffer at the exact same speed. Because if I'm bigger, all right, I'll have more charge, and so I get more pull. If I'm smaller, I should be able to move fast, but I'll have less charge. And so because the charge to mass ratio is exactly the same, they will move at the exact same rate in an electrical field with no resistance. Okay, but we're going to use resistance because we're going to make them go through the screening effect of the polyacrylamide. Okay. We also add in this beta-mercaptoethanol to break any disulfide bonds, and this does a couple things. Remember size I talked about in chromatography. It was the tumbling effect. Well, when you do SDS-PAGE gel, what you do is you unravel the entire protein. Okay? Most of them unravel, get coated with this SDS, and they all become about cigar-shaped. That's kind of what they are. And so when they tumble, they get a certain volume, and these are much, much more relatable to if their molecular weight doubles, the size of the tumbling doubles. And so doing SDS-PAGE gel, we are more likely to reflect the size versus molecular weight. And so much so that we use this experimentally to kind of get an idea of how large proteins are. All right. Now, to denature the protein, we put it into a solution. We heat it up. It has SDS, beta-mercaptoethanol, and we heat the bejesus out of it. Okay? give it lots of energy. It unravels. We put that into a well at the top of a gel. We turn on the electricity, and we allow it to run. Now, the other thing is, remember, proteins and buffers are all clear. We put a dye in the mix. As the electricity runs, the proteins will run through it. The dye is so small that it's the fastest thing that runs. And to make sure we keep all of our sample on there, we run the dye to the edge, but we don't push the dye off. If the dye is the smallest thing, and it's still on by the end of the experiment, so are all of our proteins. All right, and so we do our sample mixture. Smallest runs fastest, largest gets stuck. All right. Unfortunately, if you just run your sample, you don't get as much information as you might want. We always run a control, and the control here is a ladder of known protein sizes. Okay, and that's something that when you do an SDS-PAGE gel, you always do. I like to use what's a product called Kaleidoscope, where they put all the different size proteins, but they link dyes to them. And so every protein has a different, every size protein has a different color. You can actually watch the separation happen in that one lane because that's the way they're packaged and sold, whereas your lane would be completely clear until you do further analysis. And so at least you got an idea that the separation is actually happening and it's going well because your reference lane is working well. All right, and so here we have the sodium didecatyl sulfate. We talked about the size to mass ratio. The smaller they are, the less charged. The larger it is, the more charged. And you can separate lots and lots of different bands. All right, you can actually have a very complex mixture separated into fine resolution. All right, but at that point in time, that gel never has color like that. It's always clear until you do further processing. So, you know, doing biochemistry is faith. You mix things, you cook it, and you hope. All right? And if you've taken a couple weeks to isolate your protein, and you take your protein, you set up an experiment, and you do the experiment, you put it on, and you do the gel, now you're 2 weeks down the road, and now you're hoping something actually happened that I can read and can tell what happened. Okay? How do we visualize this? We can do several things. A lot of times, part of the gel is placed into a dyeing solution. Kamazi blue is a dye that binds to proteins and sticks. And so you can put it into this really blue solution, and you let it mix for a little while. The entire gel becomes blue because it sops up that dye. Then you put it into a destaining solution, and it will leach out all of the dye that's just in the jello, except where it's bound to a protein, and you can identify where your proteins are, and you see bands where your proteins are. Kamazi blue is cheap, and we use it a lot, but it's not that sensitive. You actually have to have a lot of protein on there to see it doing that. We can do silver staining. Silver reacts with the proteins. You can get much smaller amounts recognized that way. Silver staining is kind of tricky, but that can be done. Way back when, a lot of times proteins and your protein of interest might be labeled with radioactivity. And so if you did that, it's a clear gel. You dry that gel, you stick it up to a piece of X-ray film in the dark, let it sit in the freezer for several hours to days. You take that out and you develop the film, and you can see where your protein is and isolate it. That's a way to do it. One thing that you need to know is that the separation of these can line up, and we can read molecular weights. If you take the log of the molecular weight mass and its relative migration distance, okay, you've done this in organic chemistry, the RF values of where it started and where it went, that kind of deal. You get a straight line based upon the log of the molecular weights, and using the reference proteins in your ladder, you can actually identify the weight of your protein of interest and get a good estimation of what the weight is. Okay, and so The weight standards, if you take a look, a lot of times you get in the 15% gel, we see that we have a whole bunch of weight standards here from 10 to 250. That means 10,000 grams per mole, or we call them 10,000 or 10 kilodaltons. A lot of times we use the term Dalton for grams per mole in this. So 10 kilodaltons to 250 kilodaltons. Take a look at this. If I use a 15% gel, I get pretty good separation down here in the smaller ones. But up here, the big ones are kind of close together. If I change the percentage to, let's say, 5, look at what happens. I can separate out very large ones pretty well, but I've lost the little ones. And so depending upon what the percentage you use will be the range in which you get the best resolution. You try to pick something that is close to your protein of interest so that you get the best resolution for what you're looking for. You can even buy gels that are a gradient where it starts at 5% and goes up to 20%. Those are a little bit more costly, but you can create not a full set of one percentage, but even a gradient percentage. Mobility is inversely proportional to the log. It can be used to, I just said that. I have no idea why I have that. All right. So the SDS-PAGE is used quite often. A lot of times the SDS-PAGE will be used to identify purity of your sample. Okay, so if you just break open the cells and you take a sample out, you're going to have all the protein on there. You'll see lots and lots of protein bands. But by the time you've done step after step after step, you should end up with one single band at the end of your purification scheme if you've purified your protein. All right? Sometimes we can actually couple SDS-PAGE gels and isoelectric focusing when we start looking at very large mixtures. And so first we would do isoelectric focusing, and we see separation, right? And so they're all separated by pi. But every band might represent more than one protein. We don't know at this moment in time. And then we can put it on top of a SDS-PAGE gel, which separates by size. And what you can see is the band that had a pi of this actually represented 1, 2, 3, 4, 5 different sized proteins that all had similar PIs. And so in a very complex mixture, you can have much more fine resolution. Here is an example of a real one. There's a lot of proteins on that thing, right? And so when you take a look at this, you're never going to separate them either in one direction completely or the other direction completely, but by using the two, you can get lots of isolation. That's a whole big mess. Sometimes you would grow up cells under two different conditions, experimental conditions, do this type of experiment, and then you compare the two to see if something's missing, something's more. And so you can use that kind of analysis if you're looking for larger changes. Okay. Electrophoresis. It is high resolution. I can separate very finely. It requires us to denature the protein. It creates an active protein, and it's in very small amounts. So I do not want to use this technique as a proof of or isolating my protein. Even though I could get it beautifully pure, it's beautifully pure dead. And that's not important for us most of the time. All right? And so we want to use the other techniques to do the active protein. Here's an example of a purification scheme. Okay, I don't know if they do it in Biochem Lab now or not, but this is one experiment that I have students do when I teach it. The homogenate, where you've just broken up the cells. You see all of those proteins. And what we do is we tend to load the same amount of protein in each lane. So I load, let's say, 3 milligrams in each one of those. Now that 3 milligrams out of the homogenate, where I just break up the cells, has all proteins represented. That is in the cell. Then I do a salt fractionation. Okay, so I precipitate using a percentage of salt. I get a little bit of purification, but not a whole lot. Then ion exchange chromatography. You can see less proteins there, okay? But your protein of interest, as you go from each step, Here, that's the one you want in the whole cell lysate. It is not a lot of the percentage, right? But look at this one. Compared to the percentage, it has increased. Here, the band has increased comparative to the rest. Molecular size exclusion or gel filtration. You can see we fractionate all the big ones and all the small ones, and you have a few, but take a look. Much more concentrated out of the whole. And then after affinity chromatography, we have one band that is representative of all the protein in that sample. And so that's what a good purification scheme should look like. And quite frankly, what a purification scheme really happens. Usually it takes 4 to 5 steps to get your protein pure. All right. Got a pure protein. Woohoo! All right. All that work, so now you can begin your experiments. So what are the things that you can do? All right. If you are isolating a protein that you're not exactly sure what it is, okay, and so you might have done a differential. 2 different conditions, and this protein was absent in one but present in the other. That protein I want to take a look at. So you isolate it. Don't know what it is, but I know under these conditions it's absent or present. Well, I need to get some information about it. I'd like to try to understand its amino acid composition, what amino acids are there. I would love to know a little bit about its secondary structure and tertiary structure if I can, and I need to know the primary sequence about what that is so I understand what that protein is. So let's do some experiments to get that information. The first thing that you do, okay, and even if you know your own protein, okay, this will confirm how pure it is. What you're going to do is get the amino acid composition. So if you know your own protein, you know exactly what percentage of tryptophan is there, and what percentage of lysine is there, and what percentage of all of these. So you do an analysis. You want to know how much of each amino acid by percentage. Well, that means I've got to get them back down to amino acids. And so to break the amide, what we do is we take 6 molar hydrochloric acid at 110 degrees for 24 hours. Okay? So, half concentrated acid, 110 degrees, 24 hours, that breaks every peptide bond. Okay? So that will break them all individually apart. Now, go back to letter codes. There are 20 amino acids, but how many 3-letter codes and how many 1-letter codes do we have? More than that, 22. Okay? Under these conditions, asparagine and glutamine have in the R group a amide bond. Under these conditions, the amides, every asparagine will be converted to aspartate, and every glutamine is converted to glutamic acid because of the experimental conditions. So in this analysis, what you're going to identify are 18 amino acids because I destroy 2 of them. I convert them into another. Okay? They can be separated, usually by ion exchange chromatography. All right? And analyzed. Okay? We can quantify them by using anhydride to change them into colors. Proline is exception to the colors. All are blue, but proline is yellow. And what I'm going to do is use a column that is, quote-unquote, standardized. Ooh, what is that? This is an important concept. Star this word, standardized. All right, I know columns can separate by charts, right? And so if I put on, let's say, 3 amino acids, I go into the lab and I pull out 3 amino acids out of a bottle, I mix them up and I give them to a tube, to you. And you put them on a column, an ion exchange column, they will separate into 3 different peaks. Okay? Well, what are they? You knew you had 3, and they separated. And when you took your separation, you saw that in test tube 5, one of them came out. In test tube 20, another one came out. In test tube 40, another one came out. Okay, I separated them. Woohoo! Which one is which? All right. Do you have any information, enough information to solve that problem? No. Okay, so Before you can use a column to identify what you have to have some references. So how do I know where each individual amino acid comes off the column? What would I do?
Compare it to its test.
You compare it? Hello. Yes, you compare it. And you compare it to standards. Okay, well, what's a standard? You go into the stockroom, you get glycine. You mix it up. You put it on the column, and you do the exact same procedure. You add the exact same amount of roughening buffer, the exact amount of elution buffer, and you take the exact number of fractions. And you find out, test tube 5, glycine comes off. All right? And then what do you have to do? What is that? You have to do it for the rest of them. How many more do you have to do? Not 21. Not 18. I've already done one. You have to do 17. Because, remember, glutamine and glutamate will never, glutamine and asparagine will never be seen in this experiment. So you have to know where each one of those comes off. Asparagine will always be converted to aspartate, and glutamine always converted to, under these experimental conditions, to glutamate. So you have to run the experiment 18 times to get reference material. Now, the procedure and the column have been standardized. You've compared them to standards and you know what to expect. So that when it comes out in test tube 5, I know everything that comes out in test tube 5 will be glycine. And everything that comes out in test tube 20 will be proline. And everything that comes out in test tube 40 will be arginine. And so, but you know where all of them come out. If you're given 3 and only 3 line up, you know what they were. But if they were at different positions, they would have been different amino acids. Hello. All right. Ooh, there's a lot here. All right, you see all those arrows? That is the standard expected place where amino acids will come out. So this column has been standardized. Okay? All right, let's think about this. There's a lot of questions I can ask about this one. Thinking about what amino acids you see and the separation. This is an ion exchange. Is the beads on these in the column negatively charged or positively charged? Talk amongst yourself. Is the beads negatively charged or positively charged in this column that we've used? This looks like a really good test question. All right. Anybody want to take a guess? You got a 50-50 chance, right? Negative, positive.
Negative.
Okay, got a negative. Anybody else want to agree with the negative? Okay, I got 2 agrees. I got 3 people out of 26 that says it's negative. How many say that the beads were positively charged? I've got 2 of that. I've got 1 that's answering both. Okay. Is your hand up?
No.
Okay. All right. All right. I got 3 of 26 that are positive. Okay. Okay. How in the world did you solve that problem? Okay. How did you solve it yourself? What did you think about in the process you thought for negative?
Well, I went down like the amino acids, like which ones are going to be neutral.
Okay, so what is aspartate? What charge is aspartate?
Negative.
Negative. Okay. All right. This is the first thing that came off. Did the negative amino acid stick? No. It got off the column as fast as possible. All right. What charge is this one? Positive. Did it stick to the column? Yeah, it stuck really, really well. It took a lot to get this thing off. Okay? So if the positive sticks to it and the negative doesn't stick to it, what charge are the B's? Negative. Okay? So, kind of a weird question, right? All right. Second question. How many amino acids are in the original peptide before we broke it apart into individual amino acids? Good guess? No? No? Felt like I had an answer there, and then no, pulled it back. Okay. Would it be 5 or 6? Probably not good on a test answer to write 4. Okay. I think you've narrowed it down to 2 good choices. Okay.
Because the sphere could also be the sphere, whatever
Which one?
that is. No, never mind.
No, I didn't hear. Okay. All right. If we've got it down to 5 or 6, okay. All right. Now you have to vote. I want to know how many people say 5. Nobody says 5. How many people say 6? Oh, I've got lots of them. All right. And then I have people that haven't voted. They failed because of inaction. All right. That could be partially right. 6 is partially right. Okay, not fully right. Okay. Why in the world is it 6, not 5? 5 is absolutely wrong. The glycine peak is how much the other ones? Twice the value. So that is directly representative of twice as much. And so you would have one equivalent of this, 2 equivalents of that, one equivalent of this, one equivalent of that, one equivalent of this. All right. Is 6 the only answer?
Is it just a multiple?
It could be any multiple of 6. It could have started off as 12 amino acids or 18 amino acids. This would only give you relative percentages. Okay. And so, there's a lot of information in that. It didn't look like it, did it? Okay? But I could now give you the percentage of each of these. And so this would be 1/6, 1/3, 1/6, 1/6, 1/6. And you have the percent composition of your protein. And so if you know your protein and you send it off for analysis, you think it's pure, the percentages should be exactly on point if it's pure. If it is not pure and there's a mixture, your percentages will be off. So that is the best way to analyze to know if you actually have a pure. SDS-PAGE is limited by how good you can see it, and so if you use Kamazi Blue, there could be 5% of protein in there that you can't see because it's in very small concentration. This analysis would actually show you that. All right. Now, amino acid composition tells you which percentages, but that's not a lot of information. It's been a lot of work. You did a lot of experimenting to get there, but that's not a lot of information. What you really want to try to get is the primary sequence. Okay? And so what we're going to talk about is in chemistry called Edman degradation. All right. This now has been automated. You put the sample inside an instrument, and the instrument adds solutions and lets it cook and drains it and analyzes it over and over and over. When they first did this, this was an organic lab with set funnels and all sorts of different things, and so it used to be really long. Now it's pretty standard. What we're going to use is phenyl isothiocyanate as the reactant. By the use of this particular compound, we can remove one residue at a time And what we're going to do is make a derivative of the amino acid. And so each round of reaction, I shorten it by 1. And then I can shorten it again by 1. And shorten it again by 1. And so what happens here is you place the amino acid able to react with the the isothiocyanate. Now what happens is, normally in analysis, I'm going to attach this to a bead, okay? And so I can drip in the reagent, it kind of mixes around for a little while, then I open the stopcock, the entire solution runs out, and your reactant's still on there because it's on the bead, and you can wash it, and then you add it again, and I can automate it. But what happens is, you're going to deprotonate this proton. This is going to attack, all right, and then I pick up a proton. Now, I've just made a derivative. That's not very helpful. But this next step is. The next step allows the base to take this proton, and then I attack here, kick up, kick down, and that kicks out. And what I've done is I've released the amino acid, and this is the R group. The first one was alanine. And I have a shorter amino acid. And I can then separate this using chromatography on a standardized column, and I know the position of every amino acid attached to isophenylethiocyanate. So I know the positions that they would come off. So I would send the solution, it would separate and have one peak, and that peak would represent the amino acid. Now what's cool is I can do this again with the same reagent. I can do it again. The chemistry is 98% effective. So if you went into every exam in your college career and you were guaranteed to get a 98%, regardless of what you did, how happy would you be? Would you be satisfied? Most of you, there are a couple in here that 98 is not enough. I know it. But 98 seems pretty good, right? All right, here's the problem. 98 is good, but not good enough. All right, as you do it, 98% get converted and you get your amino acid. You get a huge peak with that amino acid. The next time, you get 98% of 98 as the peak, okay, of the real one, because 2% was the old one, and that comes up. Well, you can tell for the second round, but by the time you get to the 50th round, only 36% would be the correct amino acid because you've got all these partials. And the partials start building up, and so the peak becomes large, but the other peaks become large too, and you start to lose resolution. You can't say this one was the biggest one because you got all this mixture. And so you can only go 50 rounds at best. Are there many proteins that are functional in your body that are 50 or less long? So this can't be used to sequence our entire protein just yet. We will talk about how to sequence bigger things